Hello to everyone participating virtual EAP meeting. My name is Ivana Držaić and I will present you work on genetic diversity of Croatian sheep breeds based on high-density SNP array. Sheep were one of the first domesticated animals, and with human migrations they were introduced to European territory. Two main migratory routes are known, Mediterranean and Danubian, where Croatia with its geographical position stands on later one. There are many papers dealing with global European or even world sheep breed diversity, but until recently, Balkan sheep were not included in those analyses, and we had a blank space considering sheep diversity. Just recently, Chani published a paper on the origin of European sheep breeds, where they, among other world population, analyzed Balkan region. They included 4 Croatian sheep breeds. The analysis was done in 50K SNP array, and even though counted on a few animals per breed, they positioned Croatian sheep in the Balkan cluster and close to the rest of the Mediterranean populations. In our work, we expand population size and density of SNP array. The aim of the study was to genetically characterize Croatian sheep breeds by positioning them in the Mediterranean context and by determining conservation status. To visualize main axes of genome variation among breeds and individuals, principal component analysis was used. For further investigation of genetic structure, NeighborNet based on innate distances was generated, and for history of admixture software was used. Current and historical effective population size trends were estimated using SNP software as described by Barbato. Individual animal genomic inbreeding was evaluated on genome-wide autozygosity estimated from high-density SNP data using runs of homozygosity described by Ferencakovic. Pair-based genetic differentiation between all pairs of sheep was also calculated. We have genotyped 4 Croatian breeds, Pug Island sheep, Dalmatian Pramenka, Istrian sheep, and Dubrovnik Ruda sheep, as well as 1 Macedonian Pramenka breed and European Muffin. That would be a total of 120 individuals. For genotyping, we have used Infinium ovine HD SNP chip that annotates 606,000 SNPs. After the quality control, some animals and SNPs were lost, but we finally ended with around 450,000 SNPs and 118 sheep. Additionally, we have added more Mediterranean breeds that were publicly available. We have included 2 Italian breeds, 25 French breeds, 1 Spanish, and 1 African breed. After merging dataset and quality control, we ended with around 360,000 SNPs and 750 animals. To investigate inter- and interpopulation genomic diversity, as well as genetic relationship among individuals, principal component analysis was performed. Here is PCA plots of the first and second principal component. Together they account for 13% of total variation and separate breeds by geographical position. The first component separated South French breed Rambouillet to the far left, while second separated breed by geographical latitude. From south to north separated Africa, then Mediterranean breeds were also Croatia clusters, and at last North French breeds. As seen on the bar plot, third, fourth, and fifth component together account approximately another 12% of the variation. This is why two-dimensional scale is not clearly separating breeds which can be seen in zoomed view. Therefore, we generated NeighborNet for the same database. The NeighborNet graph confirmed PCA geographical clustering, but also gave us insight into more detailed structure. Here we can see that Croatian breeds are more related to Corsican sheep and Italian breeds, but are also more distant from French sheep breeds. Using standard admixture cross-validation procedure, we detected that lowest cross-validation error was for K11. But here we also plotted K17 as the breeds continued to clearly separate. We have repeated the admixture analysis only with North Macedonian and Italian breeds, and as before, we decided that K5 was best describing population admixture. On the larger dataset, it is interesting to notice that there is no greater influence of the merino sheep on Croatian breeds, even though there are records that the merinization process occurred. It can be seen that all breeds have separated and the Dalmatian Pramenka is intermixed mostly with North Macedonian Pramenka. These two sheep breeds are also geographically closest with no barriers like sea or larger mountains in between. Also, we can see admixture signatures among Pramenka type breeds. The FST values for all pairs of populations differed from 0 and ranged from 0016 to 0068. The closest pairwise value was observed between Dalmatian Pramenka and Pug Island sheep. Dalmatian Pramenka generally expressed the lowest FST value compared to Croatian breeds. Within Croatian breeds, the highest differentiation was observed between Istrian and Dalmatian root. We have used SNAP to obtain historical effective population size and also linear model to estimate current population size. The results from modeling historical effective population size show a declining trend, especially for the Dalmatian Pramenka sheep. Comparing to other breeds, Croatian sheep have lower effective population size. Considering that sheep leverage genome-wide SNP linkage disequilibrium and that the data will tend to underestimate effective population size because of physical linkage between SNP, we have also calculated inbreeding effective population size. Using delta F for calculating effective population size, it is observed that Dalmatian Pramenka has the largest MA. This is logical as Dalmatian Pramenka also has the largest population size. Different values were obtained for effective population size as a result of different methods, but as well as a result of recently constant population growth in the Croatian sheep breeds. Genome-wide autozygosity estimated from high-density SNP data using runs of homozygosity are visualized for the individual animals from the Croatian dataset, but also for a range of Mediterranean comparing breeds. There is significant variation in inbreeding values among individuals and between breeds. We can see that Croatian breeds have relatively large recent inbreeding as well as Italian breeds, but to a contrast to French breeds. To conclude, we have seen that Croatian sheep breeds are distinguished from other Mediterranean breeds, but related to Corsican sheep and Italian breeds. Some breeds have lower effective population size based on LG, but larger inbreeding effective size. And that shows that diversity is not in danger. We have seen lower inbreeding present in Croatian breeds, but also higher recent inbreeding. This higher recent inbreeding was obtained, but it can be controlled with breeding programs. In the future work, we plan to add additional 4 East Adriatic breeds, and that would be an account of another 100 individuals, and also to include other sheep breeds from Europe and ancient samples. Thank you for your attention.